My name is Marissa Maciej-Hulme. I'm now in Oslo Met in Oslo, Norway. I moved recently, like last month, to start a more of a glycan-based approach using GAGomics to really kind of bring the glycan types together. But previously, I've been working with GAGomics, and so this kind of ties in quite nicely. I was doing this research over a number of years on and off. So we have a number of funders here, including the Dutch Kidney Foundation, Health Holland, and my new grant, the Marie Curie Actions. So, oh, sorry, it just won't change. Give me a sec. There we are. So I want to talk to you today about GAGomics, and I want to get you excited about GAGomics as a future field for emerging into current glycomics. It's been a fledgling field for a while, and I think that it's gaining a lot of pace. So in my presentation, I'd like to explain why we want to develop GAGomics. What is the point of doing the GAGomics development in the context of precision medicine, and also in the context of how we can stratify disease using different glycans and structures of glycans. Then I'm going to go through a little background about glycosaminoglycan structure and how we think glycomics can reveal this heterogeneity, which is commonly associated with cells and tissues and disease sets. And then a run through of where we're at with GAGomics, going through diagnostics of cancer towards oligosaccharides, and what I think is a roadmap for the future for oligosaccharide GAGomics. And this is coming through from a pilot study looking at plasma HS in kidney diseases. And then naturally I'll be concluding and looking at the future challenges. And this is what I really would like to have as discussion, is what people outside of the field and inside the field also think, so that we can collaborate and tackle these big challenges together. So why GAGomics? Well, precision medicine is a term that's been around for a while, but describes an overview of the omics technologies, which represent a rapidly expanding field. And this is pushing towards a holistic view of healthy and diseased tissues, whilst embracing individual heterogeneity. It relies on the identification of biomarkers, predominantly, and these are, throughout disease, important snapshots to understand the progression and the response of therapies, but it also gives you an overview of what's happening in the cell or an organism at any given moment. Stratification of patient disease groups is vital for this. And you can see here that precision therapy would rely on different treatments going for different target groups. And gene, RNA, and protein approaches are commonly adopted to stratify these patient groups, with lipids and glycans coming forth now. And the glycosylation status of a specific protein, a cell, or a tissue will be instrumental for going forward and designing new treatments and strategies. So in this field, glycans we know is an alternative way to stratify disease. We know that they are highly heterogeneous, and we know that there are many classes. Almost every protein has one or more post-translational glycosylation modification. And this involves the addition of glycan structures after the proteins, or during the proteins being made, and also afterwards. Glycans are broadly classed into N-linked, O-linked, and glycosaminoglycan GAG families, each one with one or more subfamilies based on their linkage structures or structural similarities. So biosynthesis of these glycans are synergistic and interlinked through the use of many common substrates, precursors, and enzymes, meaning changes in one glycosylation type could impact on another. We're particularly interested in the proteoglycan family, which are dominated by long linear GAG chains. Glycosaminoglycans, or GAGs, these are comprised of repeating disaccharide units and are split into 6 types. They're the carbohydrate component of the proteoglycan, and 3 are shown here: heparan sulphate, chondroitin sulphate, and dermatan sulphate. Synthesized in the Golgi, these 3 sulfated GAGs are built onto a tetrasaccharide linker, and that's anchored to a select serine residue in the protein core of proteoglycans. The non-template driven action of the glycoenzymes, depicted here and here and here, give rise to the finely patterned motifs for ligands and exert GAG functions. GAGs are heterogeneous in nature due to the incomplete action of the enzymes and downstream modification mechanisms such as heparinase, hyaluronidase 1 and 4 over the life cycle of the GAG. Numerous different protein cores also add to the heterogeneity. So why GAGomics? Well, we know that GAGomics is a very powerful tool and gives an overview of structural changes in a sample. It's particularly suited to profiling heterogeneity. Current GAGomics methods are well established for N-glycans, O-glycans, and glycoproteomics following closely behind. But what about GAGomics? Glycoproteomics has been helpful here, detailing the site of specific glycosylation of GAGs, characterizing which chain is CS, DS, or HS, and has resulted in the discovery of new proteoglycans, largely led by Noborn and colleagues. As you can see here, that they're in a review that they published 2 years ago, they've actually found many new HS and CS proteoglycans using a glycoproteomic method. However, GAGomics typically concentrates on the linear polymer attached after the tetrasaccharide linker, with various compositional methods developed using bacterial lyases to digest the chain into disaccharides. Standards are commercially available for these methods and have been successfully implemented in mass spectrometry since the early 90s. In fact, last year, a group, Bratulik and colleagues, published an intriguing paper on disaccharide GAGomics using a commercialized kit investigating the early detection of cancer using non-invasive plasma and urine samples. Across the different cancers which are depicted here, they looked at changes in total GAG type, as well as hypersulfation and net negative charge, and found that these were connected to different cancer types, which affirms this idea of glycans to stratify cellular heterogeneity in the concept of disease that I discussed earlier. Importantly, they also showed that GAG changes occurred early in disease, irrespective of cancer type. So here is the healthy cohort, and these are all the cancer types put together, and then they staged them between early stage, stage 2, stage 3, stage 4, and high grade. And you can see here that quite clearly there's a difference between the healthy controls and early stage or stage 1 of a cancer type. However, although any cancer detection is clear, which one cannot be deduced from disaccharide GAGomics? Otherwise, they would have shown this in the data. And this actually highlights one of the main problems with disaccharide analysis: oversimplification. We know, so following on from this, I'm going to be using heparan sulphate as my main example here, but it is also applicable to CS and DS and also keratin sulphate. We know that HS and other sulfated GAGs have many heterogeneous structures. And you can see here is a plethora of diseases that heparan sulphate has been associated with, not just cancer, but kidney diseases, diabetes, brain diseases, Alzheimer's, obesity, and also genetic diseases. So we know that heparan sulphate is highly heterogeneous, with different parts of the chain encoding regions of distinct sulfation patterns that orchestrate ligand binding. We also know that depending on the cell source or the tissue, can create different effects and underlying mechanisms for homeostasis, but also disease. However, some of the fundamentals of how this occurs and why remain unknown. For example, is the GAG chain always the same on any given site on a given proteoglycan? Does this contribute to the heterogeneity of the HS observed? Is the GAG chain fundamentally the same from person to person or species to species? And are all sites occupied 100% of the time? So oligosaccharide GAGomics can begin to answer some of these questions by retaining biological structures and the natural heterogeneity of samples. Some free oligosaccharides already exist from the action of endogenous heparanase. Other structures can be generated from controlled bacterial lyase digestion, if warranted, and possibly new GAG-specific enzymes that may be discovered in the future with different specificities. Since oligosaccharides often retain biological activity, probing GAG oligosaccharides with known ligands may also provide useful information for relevant glycan species. And we know that oligosaccharide profiling will retain more structural information. So how do we get there? The quest for more biologically informative information doesn't come without its challenges, and the field is in its infancy compared to other glycomics. Right now, almost all analysis is done manually. And I say this with a heavy heart, which is time-consuming and limits the number of samples that can be analysed. And this, I believe, is currently one of the biggest bottlenecks towards mature GAGomics. Public mass spectrometry data of GAGs is also therefore scarce, which blocks further analysis and cross-referencing of data to transcriptomics or use to design targeted glycotherapeutics. This leads into my second point, which looks at the limited biological sources. Data generated from different biological sources is important, and we know that GAGs are changing in many homeostatic processes and diseases. But often, if a glyco part is included in project design, NO glycomics are chosen over GAGs simply because they are a safer option to generate insightful glycan data. Oh, sorry. Especially, of course, if the samples are precious and or small in size. The trend of single cell and deciphering heterogeneity on a single level is something that cannot be ignored by the proteoglycan field. But considerable work needs to be done to make that a reality. And one of the ways that this could potentially be done is by integrating with other glycomics and improving analyses from, preparation methods from small material size. I will not talk about this further today in this presentation, but I'm happy to discuss it later and is a current focus for me in the lab. Further down the line, it will be necessary to continue solving isomeric issues of mass spectrometry, as currently their separation is tricky and very specialist. And the generation of oligosaccharide standards will also need to be completed. Their availability is limited, mainly because we don't know which standards to make. So improvement in the biosources and data generation should help guide standards, which standards will be important to synthesize and implement routinely for particular analyses. So as a postdoc in the Van der Vlag lab in the Netherlands, I was investigating the role of heparan sulphate as a novel biomarker of kidney diseases and became interested in GAGomics as a top-down method to search for potential HS-based biomarkers. The Amster group at UGA in America had developed a mass spectrometry method, one of a few, for HS and heparin using an online capillary zone electrophoresis with a custom-built ESI-MS interface. You can see here like an overview of how this would work. You have a buffer reservoir, and the voltage pushes the samples along in the CE capillary based on the net negative charge and size distribution, and then this gets directly sprayed into the mass spectrometer. And so the separation occurs before the MS spectra is collected. And they've done this for urine samples. So we decided to do a pilot study looking at kidney diseases and heparan sulfate modifications. We knew in the past that heparan sulfate it changes in different kidney diseases, and we wanted to explore it as a potential biomarker. So we collected a number of patients with 6 different kidney disease types and 11 healthy controls. We collected the plasma. We purified the heparan sulfate, which I'll go through in the next slide, using classic anion exchange chromatography methods, and then ran it through this capillary zone electrophoresis mass spectrometry method, and then found a bit of a bottleneck when we got to the glycoinformatics. So this is the overview of what we wanted to do, and we had to develop some of this technology here to go forward when we had this many samples. So to prepare our samples, we isolated the plasma from the blood, and we digested pretty much everything else away. So we did chondroitinase ABC for chondroitin sulfate and dermatan sulfate, DNA and RNA, and protein digest. And then we put the samples through an anion exchange chromatography column to separate out the intact heparan sulfate oligosaccharides and get rid of all the small digested material. And then after a series of desalting steps, this is when we performed our CEMS and took it forward for bioinformatics. Once we'd collected the raw MS data, we before we collected the raw data, we took the peak picking using the in-house software of MZMine, and we converted this using in-house software to structurally predict the length and how many sulfates on the heparan sulfate oligosaccharides that we had predicted to detect. So we created lists, and then we did this for healthy control data and for disease patient data for each disease subset. And what we then did is took the healthy control features out of the disease patient data to look at unique HS species as a first cut for looking at interesting heparan sulfate as biomarkers. So once we'd done this, we then condensed our data down. At this point, we had 3 separate runs from each patient, and we'd taken each sample to look to see if we had any injection variabilities or run-to-run variabilities. And then we actually also curated our data by looking at whether species were found in 2 of the 3 lists for each run for each patient. And if it was found in 2 or more, then it was kept on the list. If it was found in 1 run but not the others, then it was discarded. So once we condensed these features together, we ran the neutral masses through the GAGwizard software to assign GAG compositions, and then using the data extracted from this, including the neutral mass, the MZ, and the retention time, we formed a matrix to allow us to look at the data by PCA. We also looked at the data by an upset plot, which allows us to look at the individual groups for unique species, and also look at crosses between the groups for different HS oligosaccharides. So what's shared between the groups. And you can see here, there's the list of the 6 different kidney diseases, which is type 2 diabetes, pregnant patients with a kidney transplant, minimal change disease, focal segmental glomerulosclerosis, systemic lupus erythematosus, and membranous glomerulopathy. So I will use the shortenings going forward. So you can see clearly here that the most heparan sulfate oligosaccharides that were unique were found in the MG, or the membranous glomerulopathy group. And not only were the most found, but the most unique species of 59 were also found. But each group has a set of HS that is unique to them. One other interesting fact that you can see from our data extraction here is that 1 heparan sulfate was found in all kidney disease diseases, and this is a heparan sulfate that doesn't have the tetrasaccharide linker, so it's a free HS chain. If you take this data and you put it into the PCA matrix, you can look to see how similar or different the diseases are. And mostly, as you can see here, most of the kidney diseases cluster together, which is quite encouraging because, you know, kidney disease itself is different to cancer, it's different to Alzheimer's. So the fact that they're clustering is quite promising that some of these HS changes may be important for kidney diseases specifically. But what was super interesting was this subset that separated out in the MG group. And the 4 patients that separated out all have an autoimmune element. So they have autoantibodies that have been confirmed and detected within the sample. At the time, we were unaware that heparan sulfate was, you know, anything involved in membranous glomerulopathy. But from searching in the literature, we then found that there was 1 or 2 papers looking at, you know, heparan sulfate as a potential connection. And this one here, published in 2019, actually found that 2 of the polymerization enzymes, excitocin 1 and excitocin 2, are associated with deposition in the kidney of a small subset of the MG patients. And this is quite interesting because they didn't find any antibodies that are associated, you know, specifically with the proteins. But this accumulation in the glomerular basement membrane, which is where the filtration happens, seems to mark this particular group. And again, autoimmune disease was common. So there may be a link between the autoimmune component of some MG patients, and stratifying this group further might be a quite interesting way to look at HS as a biomarker. So this is going back to the roadmap that I mentioned earlier. This is where I think we're at. I think that we are very early in the infancy for GAGomics, or oligosaccharide GAGomics specifically. And I think there's a long road ahead. But I think from looking at some of the pilot data, you can see that the stratification is quite interesting to really pull apart the different groups. And from looking at the promising data that the disaccharides have shown in the diagnostics for cancer, for example, but also in more general data that has been generated in the past from, you know, disaccharide structures in immunology, in Alzheimer's, in, you know, tissue specificity. I think that there's a lot of data that could be gleaned by just pushing forward towards a more oligosaccharide approach. So just to conclude and go back through these future challenges and open up discussion, we are at the stage where we have, you know, developed a top-down profiling method for plasma now, as well as urine oligosaccharides. And we've demonstrated that with the pilot data from the kidney disease patients, but this is now applicable across many different disease types, and would be interesting to look further to see how similar or how unique some of these HS oligosaccharides are for, you know, stratification against kidney disease versus cancer, kidney disease versus sepsis, for example. And The data processing algorithms and the pipeline is now in place for us to be able to do this on a larger scale. Doing it manually, patient by patient at the beginning was really difficult. So we quickly found that we needed to have a more bioinformatic approach. And we've now generated the first curation of HS oligosaccharides from a natural source. There have been instances where oligosaccharides have been generated in the literature from other GAG methods, from various other approaches, but this one here really compares the different groups to allow us to start detailing the disease. So there's a long to-do list, and it would be first on the list to expand these HS sources and fortify the data. Really kind of take the HS oligosaccharides from biologically driven questions to sort of bring a large amount of data into the field, but also have cross references. And we also need to continue building the extensions to include GAG analysis into bioinformatics, because GAGs themselves have their own unique challenges and unique information that we can gain from them, and how we put that into bioinformatics will really make a difference to how the scores, or how people have described it in the past, how you can stratify or make a glyco score, for example. Another thing that I think is really important to take forward is this access to GAGomic technologies. There are some lovely methods about, but the uptake is somewhat slower than other glycomic methods, probably because people are shy about how complicated GAGs seem. So if we can increase the ease of the preparation methods that lead up to the GAGomics, and then access in that way to the technologies, we can fortify more collaborations. And one way to do this is to try and integrate GAGomics into other glycomics and keep it compatible with other omic approaches. For example, transcriptomics. If we could get the glycomics and the transcriptomics together, then people would not have to choose. And of course, it's always better to make things cheaper. But if we can also simplify and speed up the process, then this will increase the access and the ease for utility. And then finally, huge efforts have been made in the past and continue now to explore new MS technologies. And there's a lot of excitement and interest around this gas phase separation of isomers, for example. And as mass spectrometers get more and more intricate and fancy, new capabilities will allow GAGomics to be pushed forward into the future. So with that, I would like to acknowledge my funding sources, but also the people that collaborated and were involved in the work. So these are Johan van der Vlak and Mark de Graaf and colleagues at the Department of Nephrology. This is over in the Netherlands. Our collaborators who provided samples, Bernard Vandenberg and Ton Rabelink at Leiden University Medical Center for some of the kidney disease patients. My new Department of Life Science and Health at Oslo Met, and my mentor Nicholas Carson, and our collaborators for the MS oligosaccharide capabilities at the University of Georgia. That's John Anster's group, and students that did a lot of the manual runs, which is Janji Kim, Elijah Roberts, and Yi King Zhang.